Thanks, Sean, for the very generous introduction. And while the slides are loading up here, I'll maybe just give a bit of a background. So I'm a basic biologist, physiologist at the University of British Columbia. I've been here since 2004 with a research lab at the Life Sciences Institute. And it was really a great pleasure, actually, to meet Sean and his passion for therapeutic nutrition fit very well with my interests in biology around this area. Most of what I'll show you today is really basic fundamental biology, but it's very special to me to see that there's an impact of the work we're doing in the lab beyond the lab. So if everybody can see the slides, I'll just start forwarding. The other thing that some of you know, I'm very active. I love being on social media. So if you don't already follow me on Twitter, please go ahead and do so, and we'll go from there. So I don't have any conflict of interest that are relevant to this presentation. I'm not going to get a dime richer or poorer, hopefully, from this, but it is worth noting that over the years I've done some consulting for various pharma, obviously in relation to diabetes, type 1, type 2, obesity as well. And actually between 2017 and 2018, I was in Oxford on behalf of Novo Nordisk setting up their type 2 diabetes basic research institute. But I've left that post, and even if I was still in that post, I don't think today's topic would necessarily be in conflict with that. So I'll move right on to the most important part is, the work I'm going to show you is the work of an incredible group of students and postdoctoral fellows in the lab. So it's always appropriate to start by thanking them first. You can see some of the current members here. I'll show you work late in the presentation from Yelena. And then a lot of the work, you'll see the published names, Aria, Nicole, various others. I also should thank the funding agencies that support this work, the CIHR, JDRF, various other ones have supported this. And science now, state-of-the-art science, is really a team sport. So all of my colleagues, UBC, people like John and Sean at the IPTN, and others, we couldn't have done this work without them. So let's start off with why we're all here. Obesity and type 2 diabetes are enormous problems. This is from a relatively recent International Diabetes Federation slide. It's actually already out of date, but you can see at this point they calculated, and you can haggle a little bit over the specific numbers, whether it's 380 million or 420 million, but there's an enormous number of people with type 2 diabetes worldwide, and almost half of them are undiagnosed. And these seem like really large numbers, and the projections are even scarier. And what's interesting is I found this slide a little while ago. I'm old enough and have been around the field long enough to remember some of the earlier predictions, and I thought, well, they must just be overestimating, or they must be guessing high. And it turns out that they've guessed wrong. If they actually go and make their predictions, they've been under-predicting all these different predictions over the years. So this is truly an out-of-control biological epidemic, and it has enormous consequences of physical and emotional and economic suffering. And that's shown here. We're at a point now where the expenditure, healthcare expenditures now related to overall health, and then the percentage of that to diabetes, as you'll see over here. We're at a situation now where the healthcare expenditures continue to increase to the point where in many countries they're threatening the solvency of their budgets themselves. So this is an enormous issue. And you can see here the direct costs of the various different diseases. One of the things to realize is that even though diabetes is characterized down here in the endocrine disease, it is, and obesity are leading causes of cardiovascular disease. Here pegged, I believe these are US numbers, at 200 and some odd billion. Obesity now is the second highest risk factor for many cancers after smoking, and it's projected by 2040 to be actually the highest risk factor for cancer. Some people have coined Alzheimer's disease as type 3 diabetes, so there's links between diabetes and many central nervous system diseases. There's just an enormous diabetes, the leading cause of kidney failure, one of the leading causes of adult blindness, of non-traumatic amputations, and together these cost society billions and billions of dollars. So, what can we do about this? So I'm going to argue today that in order to solve this problem, we really have to understand the fundamental biology. And I think you can go back to, this is a quite an old picture, and I'll redraw it in a second. But what this describes is the time course of type 2 diabetes pathogenesis, starting at a stage of obesity and impaired glucose tolerance, and then ending at a stage of frank diabetes. And the lines you can see here that are of importance, here you can see postprandial glucose and fasting glucose, and here's the endogenous insulin that happens much before that as a rise. What's important to realize for this talk is the endogenous insulin and the insulin resistance rise at the exact same time at the exact same rate. And so there's been always some question about what's the chicken and what's the egg here. I've just redrawn this graph, but now I've put some numbers. Here is a recent estimate of the number of people that have type 2 diabetes. The number of people with prediabetes is probably twice this much. And the number of people who are overweight or obese globally is almost 2 billion people. And so you start to think about the numbers here. And as you can see here again, the adiposity, the insulin resistance, the hyperinsulinemia all happen very early, and the hyperglycemia trails after that. Looking at this, however, and as I mentioned, I spent a bit over a year working for the world's largest maker of insulin, and one of the largest drug companies in this space. And I can say that all of the drug companies and all of our medication armamentarium we have works at this stage. So right now, we treat the symptoms of type 2 diabetes. The drugs are effective for many people. They do keep getting better. They also keep getting more expensive. And what they do is they essentially replace the beta cell in a way, for example, with exogenous insulin. They improve the function of the beta cell. This is, for example, GLP-1 agonists. Or they can bypass the need of beta cell function. This would be SGLT2 inhibitors or things that improve insulin sensitivity. But what I can say about all of these drugs and that are on the market today is, although they treat the symptoms of type 2 diabetes, if you remove them, the symptoms will come right back. And that is to say that they are not disease modifying. So there's nothing we have now that you can give to a patient that will modify the course of the disease. If we were able to come in at this stage, if we were able to intervene at this earlier stage where there's still pretty good functional insulin responses and the beta cells that release insulin are still there and healthy, we don't have significant fasting hyperglycemia at this stage. Potentially, if you could protect beta cell function, you might be able to change the course of the disease. My lab does a tremendous amount of work in this area, but I won't talk about any of it today. If we were able to intervene even earlier in this stage, we have an opportunity, potentially in billions of people globally, to change the course of the disease. And this is the area that would fit in the category of diabetes prevention. So I would make the argument for this session that we really need to understand what's happening here. Now, as I've mentioned a few times now, these are coincidental with each other. But if you open up a diabetes textbook, or if you talk to somebody who teaches at a standard medical school, they'll tell you that the pathogenesis of type 2 diabetes starts with a diet and obesity, but it also starts with insulin resistance as a primary defect. And they'll tell you that the hyperinsulinemia is a consequence of this, a reaction to the insulin resistance. This is how we've all been taught. And people can argue a little bit about which one of these is primary and secondary, but the key thing is that many people, probably 95% of scientists and 99% of clinicians, would tell you the hyperinsulinemia is a response, is a consequence rather than a cause. However, there's a few reasons to suspect that this might be maybe an oversimplification, or that we might have it backwards. And this is a paper, I must admit, it's a paper that uses extremely complex statistical and mathematical modeling arguments that I don't quite understand. It just came out of Pune, India, a few weeks ago, but I've been chatting with the senior author here. But essentially what they've done is formalize something that has been obvious to many of us for a while, and that is that the HOMA-IR concept as a measurement of insulin resistance is essentially a circular argument. Because at a population level, if you're not doing hyperinsulinemic euglycemic clamps, you're inferring insulin resistance from the fact that fasting levels of insulin are high relative to glucose. And so what they did is they tested in several large populations, they did a statistical test to see if this could even hold true. And you can read this for yourself, but they basically say that if the HOMA indexes of HOMA-IR and HOMA-beta are actually reflecting what they're supposed to reflect, that classical pathway of the insulin resistance being primary cannot be true. And they restate it a second way down here, that these things cannot simultaneously be true. Anyone who has better statistical skills than I, please feel free to read this paper and maybe explain it to me. But I'll ask Malin to walk me through it sometime. But basically, this formalizes the idea that it's really a bit of a circular argument. I trained as a comparative endocrinologist, and in every other aspect of endocrinology, whether I trained in the pituitary, and neuroendocrinology, if you have high levels of a ligand, of a hormone, of a factor, you will get desensitization of that receptor. So in all other parts of biology, we tend to think of the hypersecretion as primary, and the resistance, if you will, the desensitization as a consequence. But what we'll do in this presentation is we'll ask the question, can the hyperinsulinemia itself be causal? And I absolutely do not want to take credit for this idea. These are actually very, very old ideas. So Professor Jesse Roth, who I know from a variety of meetings, he's been around for a long time. He was working in this area before I was born, which can give you a little bit of an idea of his seniority. And he asked, paper is not even that long ago, he asked in this paper, is hyperinsulinemia the cart or the horse? And this was a follow-up, was commentary on a longitudinal study that they did, where they found that in a 24-year follow-up, hyperinsulinemia itself was the strongest predictor of people that went on to get type 2 diabetes. So as with many human studies, this showed that there's a correlation, and actually showed that there's some predictive value. But as we all know, correlation does not equal causation. We need a bit more evidence to find out whether hyperinsulinemia is causal. So we can also ask the question, is a rise in insulin sufficient, for example, to induce weight gain? And we actually know this from basically every large clinical trial, and any of you that are clinicians and talk to patients, you know that when you take insulin, especially some of these pretty potent long-acting basal insulins, you will get weight gain. So that tells you that adding insulin to the equation is sufficient to increase weight gain, and this is a well-established thing. But does this tell you that this is that it's necessary? So that can only be the necessity of a certain factor can only be determined by what we call in biology a loss of function experiment, and that's when you reduce something and show that the other factor is also reduced. And that's actually been done, and this is some references to some pretty old studies now. This is back in the late 90s, 1990s, work showing that diazoxide, which is a drug that inhibits insulin secretion from the pancreatic beta cells, was able to cause weight loss in obese hyperinsulinemic adults. And this fit with some work they did in animal studies. This has actually been repeated a few times. There have been some other studies using different doses where they didn't see an effect, so it remained a bit controversial, but I'll just show you an example. This is out just this year in JCEM, which is a pretty well-respected clinical journal in endocrinology. You can see here a pretty dramatic reduction in body weight in the diazoxide treated group, and the patients here that were able to drop their insulin the furthest saw the greatest change in fat mass. So is this an open and shut case that we know that insulin itself, hyperinsulinemia, causes obesity? Well, not really, and that's because the diazoxide can do things in addition. It's a bit of a messy drug. It can do things in addition to decreasing insulin secretion from the beta cell. For example, there are KTP channels, which are the targets of diazoxide, that are in the hypothalamus and might affect food intake as well. So many people in the field did not view the diazoxide results. There have been similar results with the somatostatin analogs, which also inhibit insulin secretion, but those also have other effects in the cells. So this is where we turn to animal models, which is where we can really ask very targeted biological questions in ways that are simply impossible to do in humans. You have all the smoke in here, but is there, here we now need to test whether that smoke equals fire. So we asked, now coming on close to a decade ago, we started a series of experiments where we asked whether hyperinsulinemia could be specifically reduced in an animal model. And actually, at the time that we did these experiments, it wasn't actually obvious that you could do this. And the reason being is that the insulin-secreting pancreatic beta cells contain so much insulin that there was a question about whether you could reduce it and not have all kinds of other effects happening, or whether reducing it genetically, for example, would even impact the amount that's released into the circulation. So we need to test this directly. And this is a bit of a complicated slide, so I'll take a little bit of time to walk everyone through the logic here, because it's the logic that we will use for the rest of the presentation. So it's a bit of a technical detail, but it's an interesting quirk of evolution that mice and rats have two separate insulin genes. Human beings just have one insulin gene, which actually bears more resemblance to the insulin 2 gene here. But what I'll show you initially here are some results. These are knockout mice, so they're genetically engineered mice. You can see the genotype here. These mice have none of these mice have any of their insulin 2 genes. So the insulin 2 gene is gone. And you can see they're insulin 2 minus, all of them. And what we're comparing are mice with 2 alleles of the insulin 1 gene, so they get the 1 copy from the mother and 1 copy from the father, versus mice that have a single allele of the insulin 1 gene. So what we've done here is made mice where we've modulated what we call their insulin gene dosage. And that's a very fancy way of saying we've made mice that are genetically incapable of sustained hyperinsulinemia. And you can see that in this graph over here. In the control situation here, in our control mice that have 2 alleles of insulin, you can see that if you give them this specific diet, and in mice, which is a little bit different than humans, if you give them this diet that has about 60% fat, they become very hyperinsulinemic. And this is all the way out to 1 year of life. These other mice that have the reduced insulin gene dosage, they try, but essentially by 1 year, they look like they've never gotten the high-fat diet. So there's a big difference between the pink and the red here. And these mice are genetically incapable of diet-induced hyperinsulinemia. And even in these mice that have a moderate diet, a controlled chow diet, you can see that after a year, if you have half as much insulin gene, you have about half as much insulin. So a bit to our surprise, this actually worked. Now, some of you might be asking, what happens to glucose tolerance? And surprisingly, the glucose tolerance, aside from a time in the mouse's life where they're really, really growing, these are teenage mice right here, and a few of these red mice got very high glucoses. But aside from that, and you can see the glucose tolerance test here is slightly higher in this red mice. These are the mice with reduced insulin. But after they get through that phase, the glucose tolerance is completely normal. And for those that don't know, the glucose tolerance test, we've injected glucose here, and we just measure how well the body, the mouse's body, is able to regulate it back down to baseline. This is a very good glucose tolerance, and as you can see, there's no difference between the groups. Over here is insulin tolerance. Here we inject insulin into the mouse, and we see how well it lowers the glucose. And actually, if you look really close, once they get old, and I'll return to this later, these mice with less insulin, they actually look like they have better insulin tolerance, so they're more insulin sensitive. So for the purpose of this lecture, we will define hyperinsulinemia as insulin levels that are in excess of what is required for sustaining glucose homeostasis. And here is the main result of that study. Again, these mice that are genetically incapable of sustained hyperinsulinemia do not put any weight on. This red line here, they are protected completely from the diet-induced weight gain. So this means that the hyperinsulinemia must have caused the weight gain, as you can see here. So what is this difference in weight gain? Well, it's almost entirely, or it is entirely due to a difference in the fat to lean ratio, shown with this NMR imaging. We can also just take out the fat pads, and you can see that this diet makes them quite fat, and then reducing the hyperinsulinemia prevents that completely. The growth of the mice is not different between the genotypes, and none of the other organs are different in size. You can see here that in this control mouse, if you give them this diet, they get these large, swollen adipocytes. This is a section through the fat pad. And these mice with reduced insulin gene dosage are protected from this, and they look very much like the normal mice. So here you can see that the body weight differences are due to an increase in energy expenditure. And you can see this was done with the indirect calorimetry. And this is the increase in energy expenditure occurs prior to the differences in body weight. And we did not measure a difference in food intake. So we think the majority of this effect is due to differences that are not mediated by the central nervous system. We used measurement of gene expression, so looking at the mRNA, or the how many copies of some very important genes are produced in this case, the adipose tissue, but we looked at a lot of different tissues. And what you can actually see here is that some very, very interesting genes showed up in white adipose tissue, which were previously only known in brown adipose tissue. And for those that don't know, brown adipose tissue is actually a type of adipose tissue that burns energy instead of storing energy. And what happened is these mice had what's called a browning of their white adipose tissue. You can see there's an increase in this protein called uncoupling protein 1 that essentially takes the energy and turns it into heat. That's how you don't break the laws of thermodynamics. There's also an increase in the total amount of brown adipose tissue in these mice. We hear a lot about inflammation in obesity, and in fact, in the mice with reduced insulin gene dosage and reduced hyperinsulinemia, we saw a decrease in the inflammation as well. And that we found to be very interesting. You also hear a lot about fatty liver these days, and it's certainly a huge problem in a lot of patients. And what you can see here is that the mice with reduced insulin gene dosage had almost normal livers. Those are on the far right side, compared to the second set in that had all these lipid droplets in their liver. So we see a complete protection from fatty liver. We believe this is lipid spillover that is prevented from happening from the adipocytes. We're not convinced that it's totally mediated by a direct effect on the liver. So conclusion number one, if you flip this around and consider the logic, is that hyperinsulinemia is required for diet-induced obesity, in part via an over-whitening of the adipose tissue and for lipid spillover into fatty liver and other tissues. I didn't have time to show you those data. And what this means is that whereas the current model, or what I would say is the past model, puts insulin resistance upstream or as a causal factor to hyperinsulinemia, which is still possible, but in our hands and in our model, and I believe also in more and more evidence is coming clinically from human models, that the hyperinsulinemia is a causal factor in the diet-induced obesity. And we know this because we could remove the hyperinsulinemia and the obesity went away. In this first set of mice, we didn't see significant effects on insulin resistance, perhaps because we didn't leave them long enough. And so none of our studies, this is why this is grayed out, were powered or prepared to answer those other questions of the order down there. So the next question we wanted to ask is, are the effects of reducing insulin durable? So do they last even when the insulin is no longer reduced? So I'll show you another set of mice here, as it turns out, and by the way, at the bottom corner are all the published papers that if you're interested in the full-length paper, you can look it up, and if you can't find it, I will be happy to send it to you, and even answer any questions about it. These are female mice, as it turns out. It doesn't really matter, male or female, but you can see there's a reduction now in the lighter colored bars, a reduction in insulin mRNA in the pancreatic islets. This results in a reduction in insulin protein in the islets. This results in a reduction of fasting insulin. But as you can see, when you get to the 1 year time point, if you look at that pink line, it actually is the same as the dark purple line. And for some reason, that single allele, in this case of insulin 2, was able to start producing more insulin. But this gave us an opportunity to ask, even at 1 year, would there still be a difference once the insulins have returned? The bottom right graph is the fasting glucose. You can see the very first few time points, reducing insulin led to a very small increase in glucose. And this is actually what you would expect, right? The insulin is a glucose-lowering hormone. You reduce it, you would expect glucose to go up. But it's actually quite surprising, to us at least, but a very, very robust finding. These groups have 30 to 40 mice per group, which is extremely well powered for this type of work. And you can see that there's a difference between the diets, the purple and the blue diet, but there's absolutely no difference between the genotypes going all the way out past 1 year. This is a bit of a busy slide, but you can see the evolution of really no change in insulin secretion in response to glucose in the top 4 panels. The glucose tolerance in the middle 4 panels is completely the same between the different genotypes, so the mice with normal insulin and with reduced insulin, aside from the 6-week time point where you see a very small impaired glucose tolerance. This is more subtle but similar to what we saw in the other model. And then you can see the insulin tolerance, so the insulin sensitivity in these time points is not different at any time point. But what you can see here now, if you look in the top left graph, this is the body mass graph, is despite the fact that the insulin has come to be exactly the same exactly the same amount at 1 year, the improvement in body weight is still significant at 1 year. So there must be some persistent effects of having the low insulin throughout life. And you can see that whether you look at fat mass or whether you actually look at the size of the adipocytes. These are these yellow blobby things in the bottom left, and these are quantified in the bottom right. We did kind of a cool experiment here as well, and this was, we couldn't have done this without our friends at Novo Nordisk, which are the only people in the world where you can get insulin, mouse insulin from. Most of the insulin that's made is actually, of course, human insulin these days. And this is a experiment where in the hashed lines, we added back insulin in a small pellet. And you can actually see that the adipose tissue mass recovers a little bit if you look in the bottom left and if you look at the pictures as well. So this really tells you that insulin is the thing that is driving the body weight in these models. So conclusion number 2 is that in growing female mice, transient suppression of hyperinsulinemia can provide long-lasting protection from obesity. So these are great, these are great sort of proof of concept biology studies, but they don't really tell you whether reducing insulin would be a good therapeutic, right? Because these mice have had reduced insulin gene dosage their entire lives. Those genes have been modified since conception. So we decided, a very talented former postdoc in the lab decided to do a really cool trick of genetic engineering. Again, this is something you can't do genetically in people, but it really allows you to understand the physiology, which we believe is very well conserved from people into rodents, into even fruit flies and worms. Insulin basically does the same thing in all those cases. But this experiment was kind of cool. In this experiment, you can see in the little diagram, we got the mice fat first, and then we used an inducible gene deletion strategy. So it's actually a really special enzyme that can delete one of the alleles of the insulin 2 gene in an adult mouse in response to the drug tamoxifen. And you can see it does a pretty good job of that. It reduces the insulin mRNA, not as much as in the other ones, but maybe a 30 or 40%. And you can see the blue trace here, the response to glucose is smaller in the insulin secretion, so there's a reduction in insulin secretion. But what you can see in the glucose tolerance test in the far right here is that there is absolutely no difference in glucose tolerance. And we actually know that this is important. When we do models that have major differences in glucose tolerance, we don't see that there's a sort of a one-to-one regulation of lipid homeostasis by insulin. So what are the results of this experiment? The results of this experiment are after 5 weeks, you can see weight loss in the entire mouse. This is mass in grams, statistically significant. And each of these three different fat pads, the inguinal fat pad, the gonadal, the mesenteric fat pad, are all statistically smaller in these mice, whereas the other tissues, the brown adipose tissue, the heart, the liver, and the brain, are not changed at all in size. Now, insulin has a lot of different effects on fat, and this is from a review I wrote with a former graduate student of mine, Nicole Templeman. Insulin can affect hormone-sensitive lipase and perilipins and lipolysis. It can affect fatty acid synthesis. It can affect glucose uptake and fatty acid uptake. So we undertook a targeted screen to find out in these mice which pathways were being affected by insulin. And the results actually showed that the major protein affected is this protein called PTRF, which is also known as cavin. And that was pretty interesting because it's known to regulate fatty acid uptake in white adipose tissue. And you might ask, what's the relevance in humans? And the answer is there are rare humans that have a genetic mutation, a loss of function mutation in this gene. And these people have what's called generalized lipodystrophy. That means they cannot put on weight. It's actually an unhealthy form of extreme lack of adipocytes. So this helps us feel confident that this is not just a mouse relevant pathway. So conclusion number 3 from this section is that acute inhibition of insulin production is by itself sufficient to cause weight loss in obese mice, and the effects of insulin might be mediated by the gene PTRF, which is known to cause lipodystrophy in humans. So what this tells us is that if we are able to intervene at a point, at least in these studies, before we have hyperglycemia, we could potentially change the course of the disease by moderately lowering insulin, not to the point where we make our mice or our people diabetic, but we would reduce adiposity. We would perhaps reduce insulin resistance. I'll show you evidence for that in a minute. Potentially prevent the diabetes altogether. And what this also means is that lipid homeostasis and obesity is sensitive to smaller changes in insulin than glucose homeostasis is. And it's a bit of a hard concept to wrap your head around because as diabetes researchers, we always think about glucose. But actually, the biochemists have known this for a long time. The enzymes that are insulin responsive that are involved in lipid homeostasis are responsive to much, much lower concentrations of insulin, much smaller increments in insulin than the enzymes and the pathways that mediate glucose uptake, for example, into muscle. So let's ask this question. Would reducing this excess insulin improve insulin sensitivity? This gets to the heart of the central dogma, right? We are told in textbooks and a lot of our clinical colleagues are told in medical school that the insulin resistance is what causes the insulin hypersecretion. But what if the cart and the horse are in fact in the opposite direction? And this came out of Nicole's beautiful, very, very well-powered study published in Cell Reports in 2017. Again, these mice either have 2 alleles, in this case it's of insulin 2, or 1 allele of insulin 2. What I've shown here is the fasting insulin at 2 years. It's a very, very small difference in fasting insulin. On the blue diet, it's, I don't know, that looks like about 25% difference, and probably also on the purple diet. And I've already shown this graph up well into the 2nd year of life of mice, and mice, by the way, will live about 2.5 to 3 years in the conditions we keep them in. In the wild, they'll be eagle food long before that. But you can see there's no difference in fasting glucose beyond those very first 2 weeks. But what happens if you look a little bit longer? Actually, if you start to look into the 70 and 80, 90 weeks, you start to see something very, very interesting. And this shows quite profoundly that there is an improvement, a reduction in fasting glucose in mice that have less insulin. And it fits a little bit with the beautiful clinical data that Sean was showing earlier. And it doesn't make a lot of sense when you think about it. Well, why would reducing insulin lead to a reduction in fasting glucose? Well, the answer is, although the glucose tolerance is nearly identical in these very old mice, the insulin tolerance, and that is the injection of insulin at time 0, you can see these light traces, the light blue and the light purple, are below their darker counterparts. Those are both, the purple and blue are both different diets. But what this tells you is that the mice that actually have less insulin have improved insulin sensitivity. And this can also be reflected in the HOMA-IR, which I just disparaged earlier. But that can be calculated from the fasting glucose. These studies should be repeated in a hyperinsulinemic euglycemic clamp, and we're going to do that. But these mice were all kept alive, as you'll see in a minute, because they were part of a very large longevity study. But what this means is that a long-term reduction of insulin increases insulin sensitivity. And the flip side of this logic is that that part of the cause of age-dependent insulin resistance is the insulin hypersecretion itself. So hyperinsulinemia contributes in a causal manner to insulin resistance. This is the first loss of function evidence in any system that this could be the case. And I think it's pretty sound, and I think probably quite important. So how might hyperinsulinemia induce insulin resistance? Actually, this is an area that's been studied for many years, and we've recently repeated some of the studies. This is done by a talented graduate student in the lab, Howard. These are unpublished results, but we presented them at some meetings, so I don't mind sharing them with the world. But you can actually model this in the dish just by taking, these are a muscle cell line, and you can either have no insulin overnight or a high dose of insulin overnight. And here you can see in these graphs that the activation of AKT, which is a very famous insulin signaling mediator, is reduced. This is classical insulin resistance in muscle, but another side of the insulin signaling pathway that does not classically get insulin resistance, that is the activation of ERK, is untouched. And the reason this happens is simply that there are fewer insulin receptors. You can see that the mRNA for insulin receptor, the A and the B subunits, and for that matter, the IGF-1 receptors that together form heterodimers, are actually decreased. We see this also at the total protein level on the very far right, and we can even see this at the amount of insulin receptors that are on the surface membrane of the cells. So quite simply, over stimulation of these cells causes a downregulation or a desensitization of the insulin receptor at many levels. Quite simply, there's fewer receptors for the cells to take in the signal from insulin. So hyperinsulinemia directly desensitizes insulin receptors in muscle and probably other tissues. So remember I mentioned a minute ago that this really large study that Nicole Templeman did in the lab was to assess the effects on lifespan. So it's interesting because about 20 years ago, people who were studying the very simple worms, C. elegans, and the very simple model system of the fruit fly, these are very common model systems for biologists to study. And they're much easier to study longevity in because the worms only live a couple weeks and the flies only live a couple months. But in fact, using a completely unbiased approach, they found that the genes that were best at extending lifespan in these screens were actually genes in the insulin signaling pathway. And what surprised a lot of mammalian biologists and a lot of clinicians is it was reducing insulin or reducing insulin signaling was the thing that could double the lifespan of these very simple organisms. And actually, as you can see, the worms and flies, these are just the names of their genes. You can see the one gene was named age 1 because it was discovered for its effect on aging. But actually, all of these genes, this is just a signaling pathway, have counterparts in mammalian signaling. So it's exactly the same function. It's highly conserved. So organisms dating all the way back to the first multicellular organisms had insulin/IGF signaling that has been the regulator of energy homeostasis for literally hundreds of millions of years. So the question, of course, is does reducing insulin signaling extend lifespan in mammals? Well, although nobody had looked directly at insulin, there has been some evidence that these signal pathway components underneath the insulin might affect lifespan. And I'll just show you some examples here. So the insulin and IGF-1 actually have insulin like growth factor 1, I apologize, have very similar signaling pathways. And there were some studies that suggested that reducing insulin like growth factor receptors could extend lifespan. These have actually been quite controversial, so there's not complete agreement in the field. There's a much better evidence that a protein called insulin receptor substrate 1, which is one of the first proteins in the insulin and IGF-1 signaling pathways, can extend lifespan. You can see this in these graphs below. The lifespan extension is indicated by a shift to the right, so more animals are lasting longer. These Kaplan-Meier curves. It's well known that dietary restriction, caloric restriction, fasting, now we know that time-restricted feeding, a variety of these other things can very reproducibly extend lifespan in mammals, probably even up into the studies of primates, although that's a bit controversial because many of the studies are underpowered. And then there's this study here, and people are very interested in this, rapamycin, which is a drug that acts to inhibit another component of insulin signaling. There's a few problems with this study. For example, in the pooled males, you're starting to see a statistical difference before the rapamycin is put on, so I don't know exactly how to interpret that. But either way, there was a lot of this smoke around this pathway, but the question was always, what about, or the question I would ask is, what about insulin? So most people, when they think of longevity or even cancer, they always think about insulin like growth factor, but they don't think about insulin. And of course, we have this beautiful animal model now where we can directly modulate insulin levels. And here's again an example. I showed this graph before. Very subtle reduction in fasting insulin. Absolutely no difference in fasting IGF-1 at either of these two time points, or no reduction, statistical reduction. So this is the first model where you can tease apart the effects of insulin versus IGF-1. And we looked at a bunch of other hormones, and there were no differences in them either. In these very, very old mice, there was a reduction, of course, in body mass, as I showed you before, fat mass, and these were some liver samples. You can see that the mice with reduced insulin gene dosage, these are all across the bottom on that diet B. This is the purple diet. Much, much healthier looking livers without these lipid droplet spots in them. We also saw, once you got to 90 weeks, a statistically significant reduction in this marker of oxidative stress, 8-isoprostane. But what is the bottom? What's the bottom line? Do the mice live longer? So this study was the first longevity study of its kind done in Western Canada, one of only a handful that have been done in Canada. These are very long studies. Nicole Templeman has really did a heroic effort here. And this one graph I'll show you is close to $500,000. This study is about $500,000 worth of research. So what you can see here in the light blue and the light pink is that these mice with reduced insulin actually live about 10 to 15% longer on average, or if you look at aggregate of the maximum lifespan. This might seem like a small effect, but remember, this is one of the smallest manipulations that's ever been made in a lifespan study. So these are 10, 20% differences over the lifetime in fasting insulin. Something that is actually very achievable with diet and exercise and other lifestyle factors. Every single mouse was sent for, everything of course was done completely blinded. As you can imagine, we sent every mouse off for a mouse autopsy because we wanted to know if there was a specific pathology that these animals were particularly protected from. And the answer was not really. So they are just a little bit better in all the categories. But one thing that did catch our mind up in the top left here is a delay in the malignant cancers. In this case, in the one diet. And hold on to that thought. But the conclusion from this is a very modest reduction in insulin improved the health span and the lifespan of these mice. And that I thought was quite interesting. But that did open up the question of hyperinsulinemia and cancer. Now this I want to make very clear. There was a big controversy in the literature. There were some retrospective clinical signals of exogenous insulin, so that's insulin treatment, being linked to pancreatic cancer about 10 or 15 years ago. But more prospective and more detailed epigenetics has shown that, at least as of now, there is no statistically significant link between exogenous, so medicinal insulin, and cancer incidence. So what I'm going to be talking about here is the endogenous insulin produced by the body. So is there a link epidemiologically? And the answer is yes. So as I mentioned at the beginning of the talk, obesity and early stage type 2 diabetes, which of course is where you have the greatest hyperinsulinemia, are linked to the risk of multiple cancer types, and most prominently pancreatic cancer. And this makes a lot of sense, of course, because the pancreatic cancer comes from the exocrine part of the pancreas. That's the 99% of the pancreas that's not the insulin-producing pancreatic islets. But it's right there next to the islets. You would expect they have about 10% or 10 times more local insulin than the rest of the body. And hyperinsulinemia itself is an independent risk factor. And that's shown in this bottom right graph. These are both obese and non-obese patients. So even independently of obesity, patients that have hyperinsulinemia have a poorer prognosis than those that have normal insulin. But again, this is an association, and the causality relationship has never been demonstrated for any cancer. So we set about to test this, and this is work by a PhD student in the lab, Annie. This is a very complicated mouse model. So you can see up at the top here and on the side, these mice either have 2 alleles of insulin 1 or 1 allele of insulin 1. They are completely lacking in insulin 2. But they've now been crossed onto a mouse model of human pancreatic cancer. So 92% of all human pancreatic cancers have a very specific mutation in a gene called KRAS, and it is a substitution of K for D. And so these mice, we can turn on this mutation just in the pancreas. That's this gene PTF1A turns on the mutation just in the pancreas. But either way, in these mice, you can see, and we're just characterizing them here in this first panel, a pretty modest reduction in fasting insulin. It's actually at 0.059, so it's not statistically significant, but you can see that it is a very strong trend to being reduced. We care more whether it's biologically significant, which you'll see in a second. As expected, you see weight loss with these mice with reduced fasting insulin. Again, this is not a one-off observation. We have seen this in every single study that we've done. But what's interesting here is that there's, again, no difference in fasting blood glucose in the bottom left here, the FBG. And this is important because some people who are interested in the link between diabetes and cancer are blaming, they think that the hyperglycemia is the mechanistic link, is the driver. And here we have a model where insulin is modestly reduced, but the hyperglycemia is not changed at all. So it allows us to tease apart the causality there. And what you can see here, and this is measuring what are called PANINs, which are these pancreatic cancer precursor lesions. So it's very early stage pancreatic cancer. And in the mice with reduced insulin, they have about half as many PANINs, and this is statistically significant here in the orange versus the blue. And what you can see on the right in this blue box is the pancreas. This is a pancreas section from this mouse. I mean, almost the whole pancreas is taken over by these lightly colored PANINs and fibroblasts, and there's very, very little. There's only a few islands of remaining healthy pancreas. And you can see, you don't have to be a pathologist to see that the mice with reduced insulin gene dosage, their pancreas looks quite a bit more normal. So what this tells us is modestly reduced insulin can reduce the incidence of pancreatic cancer precursors in mice. And we've started to present this data at a variety of unpublished data, but we presented it at some of the top conferences now, and I think people are pretty excited. We'll write our paper soon. All right, let's move in. So that, I hope I've convinced people that hyperinsulinemia plays a causal role in a whole variety of things. I'm not saying that insulin is good or bad. Obviously, if you have zero insulin, you'll have diabetes, and that's a really obviously huge problem. But what it does tell you is that beyond the control of glucose homeostasis, you can have excess insulin, and how is that regulated? So for the remainder of the talk here, I'm going to discuss ways that I believe we can intervene, and things that initially may cause the hyperinsulinemia. So what are the factors that regulate hyperinsulinemia? So there's actually, I'll just list a few here down at the bottom. I think we are often, the way we develop in utero and as early children, teenagers and young adults, we may be, setting ourselves up for baseline basal insulin secretion that initially is adaptive, but might be maladaptive later on in life when you're more at risk for type 2 diabetes. What about how much and how often you eat? What about what you eat? What about the macronutrient composition of your diet? So I'll go through all three of these and just give a little flavor of our thinking in these areas. So development. So this is a slide here from a recent review, and it just goes without saying, maybe we've kind of forgot about this in public health, but there's an enormous amount of the type 2 diabetes risk that is already sort of imprinted before we're born and before we're grown up. So fully 50% of the risk of somebody getting type 2 diabetes is already established by the time you're 5 years old. So it doesn't mean you can't do anything about it as an adult, but we really have to think about maternal health and early childhood health and even adolescent health. But I'll just give you an example in our mouse models of what I mean by this. So in the mouse model, interestingly, this whole area of research started as our interest in trying to understand how insulin affects its own cell. So we were trying to understand how the number of beta cells, the amount of beta cells, the so-called beta cell mass, this number of insulin-secreting cells, is regulated by the amount of insulin around. And here again, in these mice, where we put them on this high-fat diet, you can see from the light blue to the light pink, we know that you get about a doubling of the beta cell area. And what we showed in that very first study is that by reducing the insulin gene dosage, you can completely prevent that. That means hyperinsulinemia helps set the amount of beta cells you have. So you have this loop, and now that can be very adaptive. The body is trying to adapt. It says, look, we've got a lot more work to do. Our pancreas has to work harder. We've got all these nutrients coming in. We need to build a bigger, stronger, better pancreas. But then when you build this bigger pancreas, your baseline insulin secretion as an adult can't be lower than the amount of beta cells you have times the basically the leak of insulin out of those cells. So you're inadvertently making a higher basal insulin later on. So the local insulin levels might help set the beta cell mass, thereby dictating insulin levels later in life. What about how often and how much we eat? Now we know that how much we eat, and these are beautiful data from the Newcastle direct study, and this is the original Lancet paper, but there's a beautiful cell metabolism paper just out this year that shows that these very robust caloric restriction diets can really reverse a lot of, this is early stage type 2 diabetes, but it's quite impressive data, and it can do so, and it can only do so when it reverses the beta cell defect. But that's the total amount that you eat. What about when you eat? So going back to 2012, these are really nice data from our friends down at the Salk Institute. This is Sachin Panda. And they did this experiment first in mice, but it's now been repeated in people. So mice will normally just eat throughout the day. They tend to eat a little bit more at night. And if you look up at the food intake here, these are ad lib fed mice. They sort of eat most of the day. They have a bit of a pulse during the nighttime hours. But their manipulation was to just feed the mice during half the day. And they showed that the mice actually ate the same total amount of calories. So it's isocaloric as far as they could measure. But what happens, you can see in the lower left here, is that comparing the red versus the pink, actually it's the opposite color, but the same general color scheme as our 2012 cell metabolism paper. We didn't call each other in advance, but I think it's kind of funny. You can actually see that the fasting insulin is dramatically reduced in the FT group. That's the high-fat diet but time-restricted feeding. And you can see these mice now showing the body weight are very much protected from this diet in terms of body weight. Does this work in humans? Well, you can't do these. This is a pretty long-term study. It's going to be hard to do in people, but they've started, and this is from a 2018 paper where they told people they could either eat for 12 hours or just for 6 hours from 8 a.m. to 12 p.m. And they showed that they ate the same amount, but what you can see in this bottom right graph is that the people that had time-restricted feeding had improved insulin sensitivity, as evidenced by the lower levels of insulin that were released in response to the glucose challenge, if you compare the two. So that's, I think, really interesting, and more work needs to be done there for sure. But meal timing, meal amounts, caloric restriction are all important, but the daily fasting and the meal timing can have calorie-independent positive effects on insulin sensitivity in rodents and in human beings. So what about carbohydrate diets? I actually, will be honest, I had very little interaction with nutritional folks before our insulin work. I've actually always, really liked sushi and cheese and stuff. So maybe I was prone anyway to having a relatively low carbohydrate diet. It's sort of what I like to eat now. When I was more of a high-level athlete, I ate whatever I could find, but I'm no longer a serious athlete. So that's I guess a minor disclosure that I love sushi, and part of why I love being in Vancouver. All right, so many of you know of these data. I find them quite impressive from our colleagues in the telehealth company Inverta. So I'm not an expert on the clinical trial, but from what I understand, the intervention here is carefully monitored, lots of communication, ketones monitoring, and if the people here adhere to this low carbohydrate ketogenic diet, they have very impressive weight loss and a remission. And these are people who are coming off of insulin and other oral hypoglycemic drugs. This to me is very impressive. As I mentioned, I have a bit of experience in large pharma, and I can tell you if this was the result of a clinical trial for a drug, this is the profile of drugs that are $10 billion a year blockbusters. It's impressive. I think this needs to be more in randomized trials need to happen, perhaps using a adaptive or some cluster randomization. I'm not a clinical trialist, but I'm assuming the smart people in the room will expand on this and really do the key trials that need to be done. And then hopefully the IPTN, we can be a part of this, and Sean showed the preliminary data. It's tantalizing. I can't wait to see the rest of the data, but we are basically seeing what looks very similar, maybe even to a more extreme level. So the question is, will this approach work for everyone? And this is actually a really good question because, I've gotten into the internet a little bit. I go on Twitter because I want to interact with real people who have an interest in nutrition. There's lots of people out there that have tried this diet or that diet. They're very, very passionate people that have found a diet that works amazingly for them. But there's also some people out there that tried a certain diet and it didn't work for them. Maybe they even gained weight. And if you actually look at some large-scale diet studies, you see a lot of variation to the response to diets. And as a biologist, as a fundamental biologist, this is really curious to me. And I'll tell you now about a project, I'll finish up the talk, about a project where we're trying to understand if there's a biological underpinning for some of these different responses. And I'm not the first person to think of this. There's this really nice study in Cell, and you can read the whole study, but the take-home message, this is just looking at carbohydrate responses. It's by Zevi et al. And this graph, basically each of these dots is a response in time, different people, and all you have to know is that this cloud is a mess. So people respond very, very differently. If you look here on the right side, there are these different clusters of people. I've worked too late here, so my auto light turned off. Okay, so some people will respond with a high response to a direct glucose challenge that's different to bread, that's different than bread and butter. Other people respond strongly to bread and less to glucose. There's enormous variability in the response just to carbohydrate. What if we take this to the next level? So glucose is the main driver of insulin secretion in most people, but it's actually not the only macronutrient that can stimulate insulin secretion. And most of the studies in this are actually quite old, but we do know that in some conditions, lipids might be able to stimulate insulin secretion, and there are several amino acids that are actually quite robust stimulators of insulin secretion. So we wanted to conduct a study to ask across a huge array of pancreatic islets. So these are islets from human pancreatic islet donors. Unfortunately, these are cadaveric deceased donors. But we have access to this material, living islets, to perform studies to ask the question, do different individual islets respond differently to the different macronutrients? And this type of study has never been done before, certainly not at this scale. We're planning, right now we're at up to about 50 participants or 50 individuals, and we plan to go to 100, 150, maybe 200 to really get a picture, sort of a population-wide picture of the response to the different macronutrients. And what I'll do over the next few slides, just so show you some interesting examples and then some summary data. So let's look at the first example. You can see the data. This is pancreatic islets. Almost all of these are courtesy of our colleagues in Edmonton. They'll ship them down to us. And this person here, male, 54 years old, was reading the textbooks before they unfortunately passed away. But they have that textbook response. They have a response, insulin secretion response, if you look at the far left, to a high glucose, 50 millimolar glucose, and then it goes back down, and then to a lower amount of glucose, 6 millimolar, it's a smaller response, then it goes back down, and then directly turning on these cells with high potassium chloride depolarization has this classical peak plateau response. And this batch of islets, now these are done side by side, so these the islets, one set of chambers is getting the glucose, and then if you look in the middle panel, there's actually quite a robust response to the amino acid leucine. Then if you put on this moderate glucose at 6 millimolar glucose, the leucine has a response on top of it. This is a very stereotypical textbook response, and then you see a nice response to potassium chloride. Again, the textbooks here show that this mix now of fatty acids, so this is an oleate palmitate mix, absolutely no response. So this is kind of what some people expect to have happen. And this is sort of the majority, as you'll see. If you were taking, for example, a diet that had a lot of fats in it, in most people you would expect less insulin secretion in most adult human beings. And this is exactly what these human islets show. No response to fat. Although interestingly, once they've seen fat, that response to moderate glucose actually seems to be quite impressive. And then interestingly, there's no real response to fat. Maybe it even goes down in the presence of glucose. And then the direct depolarization is you've seen here. Well, what about this person here? Here, this person has not been up on their textbook. So this is not what you would expect if you read the textbooks. You've got a nice response to high glucose here and a nice response to moderate glucose. You can see this in the far left panel. In the middle response, in the middle panel, beautiful response to leucine, and then leucine in the presence of glucose. But look at this. This person's islets have a response, actually pretty reasonable response, in response to the lipids. This is the exception. It's not the rule. I'm not trying to overturn all of biology here, but it tells you that there are people, or at least at this stage, people's islets. We will try to repeat some of this in a clinical study once we have enough preclinical data, that actually have a reasonable response to fatty acids. And if you told this person, oh, you need to have a very high-fat diet to keep your insulin down, you'd probably be giving them the wrong advice. Same thing with this person here. In this case, the response to the fatty acids was actually the same as the response to the leucine. The response to the fatty acids was not found in the presence of high glucose. So there are all these different permutations of responses, an incredible variety that I just don't think had been appreciated before. And you can see here, if you plot the insulin response to leucine versus glucose, it's actually pretty correlated, but not that correlated if you do the oleate palmitate in that middle plotted against the response to glucose, you can see these high dots are these outliers, these rare people that have very high responses to fatty acids, to lipids, quite low responses to glucose. Again, if you told these people that they need to have a lot of fat in their diet to keep their insulin down, again, assuming it translates into intact people, you would be giving them the wrong advice. These are just some comparisons to age, BMI, and hemoglobin A1C. And what you'd see is there's not really any strong correlations. So what we're doing in collaboration with our colleagues in Oxford is we're basically doing genetic analysis of all of these islets. Because what we would like to find out is, are there specific genes, genetic markers, that dictate these subgroup of islets that respond high to fats or higher to amino acids or higher to carbohydrates, that you could theoretically do a genetic test down the road, take a cheek swab, and we're talking years down the road, and tell these people, you know what, this is the ideal diet for you because you've got, I don't know, a fatty acid transporter in your beta cells that means they're going to see a lot more fatty acids or some sort of signaling thing. There's a lot of basic biology that's left to do, but the conclusion here is that individuals have highly variable patterns of insulin secretion and macronutrient responses. I believe that personalized therapeutic nutrition is the future, but I believe also that we're not there yet. I know that there are companies out there now that will sell you services based on some genetic markers. I don't think that we know the genetics well enough and we understand the physiology and the basic biology of insulin secretion well enough to really give informed advice. But when we are there, I hope that the IPTN is really leading the way. So I will conclude with a model of the of where I see the pathophysiology of obesity to impaired glucose tolerance to type 2 diabetes. This is a figure I took from an article that Jake Kushner and I wrote for the endocrinologist just this year. If you can see up in the top corner here, the entire environment from meal size, meal timing, meal composition, activity, I didn't even get into that, conspire to drive basal hyperinsulinemia as a condition. This promotes insulin resistance, and then of course you have a vicious cycle here. I'm not saying that insulin resistance is also not a target of the environment. I just don't think that is always the initial driver. We know, for example, that stress can have direct effects on insulin resistance, many other factors, but all of those things conspire to stress out a variety of different cell types, including the adipocytes. You get ectopic lipids that go into a lot of places where they don't belong. They leave these stressed out adipocytes once they've reached an adipocyte storage threshold. They end up in places like the liver, the skeletal muscle, the heart, the pancreas itself. And then if you start to see the curves, the insulin secretion will start to fail. And this is on the background of the genetics of type 2 diabetes. Many of the genes that increase the susceptibility of type 2 diabetes, we know these are beta cell genes, and they directly regulate the adaptability, the flexibility of the beta cell to handle this stress and impair glucose-stimulated insulin secretion. Once you have type 2 diabetes, I think it's very important, in addition to glucose-lowering drugs, which I think are obviously appropriate for some people, I think we need to really think about lowering the carbohydrate load, and either in conjunction with the drugs or instead of the drugs, if people can do this. This is a lot to think about, and these are my final conclusions, and then I'll take questions. But suffice it to say that the current treatments do not permanently modify the course of type 2 diabetes. I think we have very strong data now, and I won't get into it because it's other people's data. I believe it's also the case in the data that's published and emerging in humans. But a moderate reduction in insulin is sufficient to induce weight loss, improve insulin sensitivity, reduce liver fat, even reduce cancer susceptibility, and extend lifespan. Hyperinsulinemia can downregulate the insulin receptors. This has actually been known for a while, but we're working on the molecular mechanism. So therefore, reducing insulin, either pharmacologically or via lifestyle interventions, may be a viable way to treat obesity and prevent type 2 diabetes. But donor-to-donor variation in macronutrient response must be considered before we can fully realize the potential of personalized therapeutic nutrition. So thank you all for your attention, and I'd be delighted to take any questions.
Thank you so much, Jim. That was fantastic. A really good reminder of the importance of just understanding the basic biology and what's going on. And what I think is really cool is that your research really helps to reinforce some of the messaging around being cautious in making generalizing or one size fits all types of approaches. So it really supports a lot of the messages that we've been promoting, which is you have to really understand these things at a basic kind of biology perspective and look at it from a physiology perspective, but also be able to look at things from the standpoint of what are the outliers. So maybe an intervention might work for the vast majority, but there still are outliers that we're going to have to look at. Okay, what are the unique characteristics that require us to look at it from a personalization perspective? And I'm looking forward to doing that research, with yourself and others to really kind of fill in some of the gaps that we currently have, because we really are just scratching the surface and that sort of stuff. I would be remiss if I didn't ask you a very simple and basic question, and that is if you can speak to the physiological differences between mice and humans, particularly as it relates to the use of high-fat diet, because I know that that's one of the biggest confusions when people are looking at the studies and trying to relate mice physiology to human physiology and the different responses in terms of how our beta cells react to the different macronutrients. And so it'd be great if you would speak to that one issue to start, and then we'll get into some of the other questions.
Yeah, that's an outstanding question, Sean, and it's something that comes up a lot, especially as I speak to more clinically oriented audiences. So as I mentioned in the talk, the insulin action and the fundamental biology of insulin and how it acts is actually extremely well conserved, from humans to rodents to flies and worms. However, the factors that stimulate insulin and the actual nutrition is actually very, very different between rodents and humans. So humans are omnivoric, obviously. We know this from the canine teeth that we have, and mice eat a much different diet than us. So the way that we can induce hyperinsulinemia via diet in mice is different, obviously, than what we would use in humans. So whereas there would only be a very subset of potentially a very small group of people for whom a high-fat diet might increase insulin, and again, we do not know how well this holds up in intact human clinical situation. Virtually every mouse in our sort of inbred mouse colonies, you would get hyperinsulinemia with the high fat. And it's a chronic effect as well. So I think that's a real key distinction there. The way we get hyperinsulinemia, the way we model it in mice, is much different than what you would have in humans. But then the outcome of the insulin and what insulin does to lipid homeostasis, to lifespan, to glucose transport in muscle, these are incredibly well conserved pathways.
Great, thank you. What I will do is actually open up, if anyone wants to ask any questions verbally, just go ahead and open your mic, and I will call you out. While that is, while people are queuing up to ask questions, I'll just go ahead and ask some of the questions that came in during your talk. So one of the questions came from Eva was, I'm confused about the white and brown fat. Can you review this from the slide before hyperinsulinemia prevents fatty liver? Do we know why the white fat is dominant? And the exercise, and the exercise info confused me a bit. So there's a slide that you were talking about the
Yeah.
browning of white fat and all that kind of thing. So go ahead.
So this is a pretty new concept. It's been around for maybe about 10 years now. We were one of the first groups to show hormonal regulation of this aspect of white fat. And actually, getting to what brown fat is another very interesting difference between mice and humans. So mice actually have quite a bit of brown fat. Brown fat is how mice stay warm without shivering. Humans, most of the time, gain warmth by shivering, and it's actually their muscles moving. 20 years ago, even 15 years ago, the textbooks would say that adult humans have no brown fat. And actually, we know that this is not the case anymore. It used to be thought that children have brown fat as a way to stay warm, but that disappeared in adulthood. It's known now that there are very small depots. They're actually around the scapula and around the spinal cord and the neck, in the back there, in adult humans. It's pretty small, so it does play a role in energy homeostasis and in temperature regulation to some extent, but it's not clear yet whether it's a real druggable target. But so the brown fat, it's brown because it has a lot of mitochondria in it. It looks brown under a histological examination, and the mitochondria are actually broken, essentially. So they are uncoupled. So instead of making ATP, the energy currency of the cell, they waste ATP to make heat. So they're uncoupled. And that's in a mouse, it keeps the mouse warm. In a newborn baby, it helps them keep them warm. And it contributes a very small amount to that in an adult human. But it uses also quite a bit of energy, and it can alter the energy expenditure side of the equation. Now, the browning of white adipose tissue is another thing altogether. So white adipose tissue, what we normally think of as fat, whether it's on our thighs or that's the subcutaneous, or whether it is in around the organs, that's the visceral white adipose tissue. Those are quite different physiologically, but the job of white adipose tissue is to store energy. However, the browning of white adipose tissue would then theoretically just open the gates a little bit to burning a little bit of that energy. And what is the normal state of white adipose tissue in a human? It's a bit of a semantic argument how brown or how white it would be. But our mouse work suggests that insulin is a regulator of the browning/whitening state of the white adipose tissue. I hope that helps clarify a little bit.
Great. Related question. Oh,
this is water, by the way, in the goblet, just
in case anyone's wondering. Did they measure, one of the related questions, did they measure any of the hormonal responses like glucagon and somatostatin? In the longevity study, we've done a little bit of that. The question is, are you talking about the islets, the human islets response, or in the mice? That's the question. So I can answer both of them. In the mice, we've done a little bit of studying of that, but we haven't thoroughly characterized the glucagon or somatostatin effect. We had the longevity. We looked briefly and saw no differences. That was mentioned on one of the slides. So we do think insulin is the primary factor here. It's the thing we manipulated. So it's everything must stem from that. In terms of the human islet macronutrient response, that would be an interesting question too, and it's one that's on our radar. Human islets actually have a fair amount of alpha cells that release glucagon, so right now we're optimizing some kits to measure glucagon in all of those samples.
And we don't have any data yet, but it's something that's of interest to us.
Great. Related to the mice who had improved insulin sensitivity, did they weigh the same as the other mice? And did they have the same activity levels?
Yeah, that's a great question. So this is where you actually still even in a detailed mouse study end up with things that are inextricably linked to each other. So the mice with reduced insulin production in all of our studies weighed less, and they were also had improved insulin sensitivity. So we cannot know right now whether the improved insulin sensitivity in vivo is because they weighed less, or they weigh less for some reason because they have improved insulin sensitivity, or whether the insulin's effect affects both those things independently. What we can say from our tissue culture experiment that I showed a few slides on is we can show that hyperinsulinemia has direct effects on the insulin receptor levels in tissue culture. So we know that that's independent of obviously the rest of the mouse and the body weight. So my prediction, my bias at this stage is that the insulin effects are have independent effects on insulin sensitivity and on body weight, but that there's also additional linking effects that can't probably be teased apart in the integrated mouse model system.
So complex. A quick question in relation to time-restricted eating. Would it make difference of eating time if the eating time was in the morning versus supper time late in the day? And maybe you can speak to the circadian, insulin release and how that affects blood sugar levels. And I don't know if that's something that you see in the mouse model, but it definitely is human.
Yeah, so that's an outstanding question as well. This is not work from our group, but work from Sachin Panda, Richard Bergman, other people who study circadian rhythm. It does seem to matter. So there's a few interlinking effects there. The length of the fast, so time-restricted feeding should theoretically have benefit no matter when it is by having a longer fast in the time you're not eating. Potentially allowing the insulin receptors to sort of recover and resensitize. Layered on top of that, there are these circadian and diurnal rhythms of fatty acid levels, of the whole metabolism that is driven by the circadian rhythms. The circadian rhythms, in turn, are actually driven by the metabolism and when you have your meals. So you could theoretically, if you lived in a completely black box, you could re-entrain your circadian rhythms to whatever you wanted. And you can do this in mice, and if anyone wanted to really torture themselves, you could probably do it in people. But the meals actually help entrain the rhythms as well. But to make a long story short, it probably does matter in our society. For the average person, it is probably better to have your meals earlier and to not eat, certainly not a big meal just before bed, because your metabolism is different late at night. This is, we're not mice in that we live in a society, and it's also very important to understand that obesity and type 2 diabetes are extremely multifactorial. We live in a society where dinner is a thing, and feasting and business meals and going out to dinner and dates, and we live in a society where there's a certain amount of privilege that comes into play when you can decide when and where and what and how to eat. We won't conquer global obesity without conquering the inequalities in our society that dictate, that allow some people to decide when, where, and how to eat, and other people not to. So I fully recognize that, stopping eating at 2 o'clock like they did in that one study would be really hard to do on a society-wide level, but if people can do it, the biological rationale is there, and maybe we gradually as a society change to having big lunches and just snacking on olives at the pub or something like that instead of dinner. Who knows?
I like the idea of the pub. One last question, I think, just in relation to a microbiome. There's lots of talk about the relationship between microbiome and development of type 2 diabetes and different types of bacteria. Is there any research that you are doing in mice models that would either support or refute some of those claims?
Yeah, so the microbiome is a fascinating area of experimentation. I'm a keen observer of the field. There are lots of people doing much more sophisticated mouse studies than we are in that area. What I can tell you is that in rodents, and this is another area where I think rodents and humans are actually pretty different, and it's just because of the society that I just mentioned. So rodents eat the same pellets every day, day in and day out, and they live in little boxes, a little bit of enrichment and whatnot, but they don't live in a society. They recirculate their food, let's just say that mildly, but there's a few things that make the microbiome in rodents quite susceptible to perturbations. And there are a lot of really powerful studies that have shown that either wiping out the microbiome or raising mice in germ-free conditions so they have no microbiome, or transplanting the microbiome from one mouse to another can have profound effects in mice on insulin sensitivity and glucose tolerance and obesity and things like that. So I think the biology is there. However, I think a very good biomarker potentially of what your gut status is and your metabolic status is. The question is whether it's a druggable target. And my own reading of the literature and some work that I've seen, I've tweeted about it a few months ago. There are studies now, there's a couple in cell metabolism where they've taken humans and made manipulations of the microbiome, either transferring the microbiome from one to another. I won't describe how that's done because it's dinner time in lots of parts of the country. But the effects on the metabolic physiology are really much more subtle than what you see in a mouse. Similarly, if you sort of use very powerful antibiotics to wipe out the microbiome, probably the most minor effects on an insulin sensitivity judged by these sort of gold standard clamp methods. So the microbiome is interesting. I think it's a great potential biomarker. There's lots of people who know much more about it that are very interested in it. And I think over the next couple of years to decades, we'll really get a handle on the human microbiome and how druggable it is. The situation in humans is a lot more, there's a lot more noise. So I can tell you from personal experience, I can change my microbiome by a trip to the pub and having some real English ales or a really spicy Indian vindaloo. My microbiome could turn over pretty quickly. And, the mice just don't have that level of variation. So any ability to manipulate that has to overcome how variable it is in humans. As I said, it's potentially a good marker, but I'm not convinced yet that we'll be drugging the microbiome for type 2 diabetes directly. I think many of these nutritional food-first approaches, they're going to change the microbiome. And the changing of the microbiome may have some direct biological consequences. I don't doubt that. But I would target it just for the holistic effects of it.
Excellent. Thank you so much, Jim. Does anyone have any final questions before we sign off for the evening. So just go ahead and turn on your microphone if you do.
Hello.
I see Ravi, yep.
Yeah, hi. I'm interested in understanding the reality of care and personalized delivery of care services like, food tracking and integration of continuous glucose monitoring and how that could be done. And I know Verta study, I have seen that, and I know they're doing an amazing job, but my problem is how each person can be empowered. I mean, there's a lot of millions of dollars involved in a study, but to scale it up exponentially, that's going to be a lot harder if the patient itself is not empowered with the knowledge of their own response to their own food that they take every day.
Yeah.
So tracking food or tracking the beta-hydroxybutyric acid and getting a health coach is a different thing, but trying to do this in an integrated manner with active daily advice or a ping from their own system. I mean, those are the things. I mean, do you guys have any knowledge of similar thing being done?
So, you've hit upon the million-dollar questions there. So, I think one of the reasons that the intense tracking and the coaching and the feedback works is because you have an element of feedback and empowerment and almost a gamification of people's journey through improving their health. What I think we will need someday, and maybe someday CGM is routine and it's less invasive, and of course Google and others have talked about contact lenses, and who knows, someday some disruptive tech will make measurement of some of these key metabolites and biomarkers, and in a perfect world you would be measuring insulin actually, because it's, I think, better than measuring glucose, but maybe measuring the ketones or whatever that personalized diet someone's put on. There needs to be technology innovation to make it such that these are that they're accessible to the general population. And right now, it's really not there. And I'm not a technologist expert, so someone in that sphere will step up. They will disrupt the space, and it will come. I hope it comes soon. But I would love to see a world where people could really see the physiological outcome of what they're eating in some way other than how tight their belt fits or whatnot. But it's a great question, and I wish I had an answer. I think the beginnings of the answer are in some of these big CGM studies and the ketone monitoring approaches. There are technologies that are in the pipeline for more readily measurable insulins and C-peptides. And then there may be biomarkers that haven't yet been discovered, circulating biomarkers that could be measured less invasively, that might be better surrogates than anything we know today. And I can just say that we really need more basic research to get to the bottom of this. But I'm happy while people are still here. I'd be very happy to take questions on Twitter or in any other format as we go forward.
Thank you so much, Jim. It's the top of the hour now, so I think we'll end things here. Once again, just our true appreciation for not only the information that you've reviewed, but also just your willingness to share with the group, not only in the form of this group that participated, but all the folks who are going to be able to watch the webinar after the fact with the recording. So thank you so much for your commitment to the IPTN and for your ongoing incredible research. It's truly a pleasure to work with you, and with that, thank you, everyone. Hope you have a wonderful evening, and I will have this posted within the community within the next couple of days if you have something that you wanted to review. So Jim, thanks again, and we will be talking soon.
Thanks for having me, Sean. Take care.